So Scott Lewis from St. Louis University will also show us an analysis of the forensics challenge, and I look forward to your talk.
Hi everyone, I'm Scott. I'm a master's student in bioinformatics and computational biology at St. Louis University, and today I'll be showing you a little bit of my group's findings for the CAMDA Metagenomics Metasub Challenge. I'm really excited to be here. We had a lot of fun going through these data, and I'm looking forward to presenting this information and discussing it with you. Okay, so I'm going to walk through this outline. So I'll first tell you about the pre-processing, or lack thereof, that we did. So as David told us earlier, Metasub does a pretty good job of cleaning up all the adapters and trimming out noisy reads. So we did very little of that. I'll go into what clustering we did. Then I'll tell you a little bit how we designed our model. And then I'll show you the metrics that we used to evaluate that model. And finally, I'll tell you about a few things that we think would be really interesting to pursue in the future. Okay, so I'll just first go and show you the metadata very briefly. So we were given all of these labels. There are a few more things that we could have included in the analysis, such as geolocation or latitude and longitude. But for this, we just used the city names and continent. So the analysis was conducted at both the city and continent level. And I'll just reiterate these questions as they were mentioned earlier, but the two things we wanted to answer with this analysis is, can we predict geographic origin of a metagenomic sample even when no reference samples from this location are known? And then how reliable are these predictions actually? So first, for the pre-processing, we ran FastQC for all of our samples. We found that generally all base positions were in very good quality, so we decided there was no need to do any trimming or pre-processing for it. So next we wanted to look at the overall cohesion and dispersion of our samples at both the continent and city level. So you'll notice on the left, in both cases, these are both very widely dispersed. So there's a lot of variation between samples regardless of origin. But you'll notice on the left, the two Gondwanan continents of Australia and Africa are both clustered and show greater global separability from all the other samples. And on your right, you'll see that at the city level, there's a lot more variance and dispersion between these, and the clustering is not so tightly neat. But we do see some independent clustering at Sacramento, which I'll remind you, that was the one city that had only one forward read, and it was not paired end. And we also see tightly clustering in Ilorin in Nigeria. So for our model selection, we used all of the OTU output from Metaflan 2. We were using these as our features to feed into our model. So given this abundance of data, it was very, very tempting to feed it all into a data-hungry model, like a neural network. But for this, given previous benchmarks and some of the results in previous CAMDA competitions, we decided that we would use a more simple random forest model. So more specifically, we wanted to make the most of the random forest model, so we conducted a guided grid search for optimal hyperparameters. So specifically, what made this special is the max depth of our random forest for all of the trees was 20, and our number of estimators was 300. So I'll go straight into the results. At the continent level, all of our continent predictions had F1 scores greater than the 90th percentile, and our overall accuracy was approximately 98%. If you look over at the confusion matrix, the most error comes from confusion with Europe, and specifically, South America was confused for Europe 3 times. And this will come in, and I'll tie this in later with the mystery samples. So we next wanted to see if we could predict the origin of cities. So if we look at the F1 scores for these predictions, none of our F1 scores were below the 80th percentile, And we achieved an overall accuracy score of approximately 95%. So in this part of the analysis, a lot of the errors attributed to the European cities. So the European cities showed a lot of variation, and when they were misclassified by our classifier, they were confused for other European cities. I thought that was kind of interesting. So now we wanted to, before we even had the sample labels from CAMDA, we wanted to see if our model could just give us a distribution and predict what the breakdown of the mystery samples might be. And so this was our prediction before we'd received the labels. This is our prediction after we received the labels. So Europe makes a lot less, much smaller portion of these pi. And also North America is completely absent from the samples. So going into our mystery score, the big reveal. Not so good. Because it's a multi-class classification problem, it's still better than a coin flip, but our overall accuracy score was 47. So two things with this. Looking at the confusion matrix, at the bottom panel, you can see that South America is confused for almost every continent. That tells us that there's a lot of variation in the South American samples, and that could be more interesting to follow up on in the future. The other thing is that there is an inherent bias in the data because the European cities and samples were much more represented. So that is reflected in our model as well. So you can see that a lot of different continents are classified as Europe. So we wanted to see what kind of things that we might plan to do in the future to make this model more accurate. So one of the things was how can we prune out features or select for the most meaningful features. So we looked at our random forest, and we found that 1,177 taxa have an importance score greater than 0 for the continent predictions. And then we found that 1,162 taxa have an importance score greater than 0 at the city level. So we think we might, in future analysis, select for just these features and see what kind of results we get. Additionally, as I described in the previous slide, we'd like to remove the non-descriptive features from our OTU tables to improve our performance. We'd also like to explore additional models, so maybe we'll follow through with that neural network model. There's quite a bit of data, so it might benefit from that. We'd also like to look into using a kernelized support vector machine because those are very good at making those nonlinear decision boundaries. And then we'd also like to integrate 16S and whole genome shotgun sequencing data when it might be available for all cities and all samples. So with that, I'd like to thank everybody on my team, everybody that helped along with this project. I would also like to thank CAMDA in partnership with the FDA for the travel fellowship and inviting me to be here. Thank you.
Thank you very much for your talk. Do you have a poster that you can send people to?
I do not have a poster.
Oh, you should have. Okay, thank you very much. We move to